Okay, so welcome. So unfortunately, our talk today begins with cancer. And cancer is when an abnormal group of cells start growing in a way that they're not supposed to grow. And the real problem arises when this cancer hits the road from its original site. And this is called the metastasis. Because you can, for example, have the cancer growing in you for 30 years and you wouldn't even know it. But the real bad part happens when it ends up spreading to other organs that it really becomes a large problem. Now, studying metastasis by themselves is a little bit like studying a tree. How in the world did a tree get where it is? And you wouldn't really be able to tell a lot if you looked at just the leaves or the branches. Like, it wouldn't really tell you the history of the tree. And when we're studying metastasis, that's essentially what we have. In order to be able to understand how it happened, we need to understand the history. Now, for a minute, imagine that you were a space invader. And I gave you a pile of these round objects. And I told you, some of these are seeds and some of these are not. Now, you might be a little bit confused because for the most part, they all kind of look the same, right? Like you've got, they're round objects. And if I said, try to figure out which one of these are a seed, if you're a good scientist, you would think, well, maybe if I planted them, I could then take a look and see which ones grow, right? Because the marble isn't going to grow anything. Well, that's not just the only part of the story you have to consider, right? I don't know how many of you do gardening. I'm not so great at it myself. But there are certain plants that like certain environments and other plants that prefer different ones, right? Like some plants like to be watered a lot, some like direct sunlight, others do not. And so when you're putting these seeds, they really have to be in the right environment for them to be able to grow. And metastasis is a little bit like that too. Where the cancer spreads, it usually spreads to an environment that it really likes and that it'll continue to grow. And I mention this because certain cancers like to go to certain organs. So if you have breast cancer, you may end up having it metastasize to your brain, lungs, bones, or liver. Now, lung cancer, on the other hand, typically goes mostly to the liver and the bone. And then there's some areas at your body that you'll just never have the cancer go to. Like, for example, your heart. That's an organ that rarely gets cancer spread to it. And at Johns Hopkins, which sees thousands of patients with cancer, in their entire history, they've only had 6 cases of cancer that metastasizes to the heart. Now, that's pretty impressive when you think about how many people actually have cancer, right? So what is it that attracts certain cancers to certain areas of the body? Now, the movie that you guys are about to see, The Fault in Our Stars, the 2 main characters both have cancer. Hazel over here started off with thyroid cancer. Now, thyroid cancer is notorious for going to either the lung or the bones, and also spreads to other organs like the brain and the liver. Now, you'll notice from the very beginning of the movie that she needs some extra oxygen to help because it definitely has already spread to her lungs. And she is at stage 4. And stage 4 means that the cancer has hit the road and it's gone to other vital parts of your body. On the other hand, her friend Augustus, he has osteosarcoma. Now, he's got to be careful because this type of cancer likes to go to the lungs, the bone, as well as the brain. He, however, at the start of the movie is in remission. Now, that doesn't mean he's cured. It just means that the cancer hasn't spread and that the cancer is controlled and that there aren't any incidences of the cancer growing out of control or even on the road. So in both cases, you've got 2 different teenagers that have cancer, and you'll see in the movie the kind of effects that having cancer, especially in the metastatic phase, how that can be detrimental to the lives. Now, specifically, what we're going to talk about today is cancers that metastasize to the brain. Now, in the previous 2 characters that I showed you, both of them are at risk of having that cancer metastasize up to their brain. And it's really kind of interesting when you consider that the brain normally is not an environment that lots of things get into. And I'll talk a little bit more about why that is a pretty unique or a pretty complicated problem. Now, the brain environment is pretty unique. It's kind of jello-y, kind of soft. It is very well vascularized, meaning it gets a lot of nutrients. And so if we were going to plant certain cancers or seeds, It would only be the ones that like that kind of environment that would really flourish. Now the problem when we're studying cancer and metastasis specifically is that a lot of times biologists use sort of these petri dishes to study cancer. And what this is essentially is a 2D environment with media on top, and media is just kind of like Gatorade that the cells like. And this is not at all representative of the very complicated environment that is the human body, right? Like we're not 2D, we have things moving all over the place, and we're not made of plastic. And these plates, of course, are made out of plastic. The other option that scientists sometimes use to study cancer is they'll try studying it in mice. And this is like the equivalent of trying to decide, or trying to understand how these big trees grow in the forest by planting them in a box. It's just not going to be the same thing, right? Because at least looking out here in the audience, I don't see any hairless white mice, right? So trying to compare us to white hairless mice is almost as silly as trying to compare this beautiful growth of trees to this little tiny box of soil. Now the soil is just one part of this problem, right? The other part is how on earth did the seeds get to where they are, right? Now we know that seeds can get stuck to like people, and that animals will eat them and then they'll poop them, and so that's how the seeds travel to a different origin. Now in our body, the way that the cancer travels to a different organ is through the vasculature. That's the main road of transportation. That's how it spreads. And the vasculature of the brain is pretty unique. You can see from this kind of cartoon schematic that some of these vessels are straight, others kind of curve a little, and some of them branch. And we don't really know whether this has an effect, right? Like, does the cancer like to go down the roads that are straight, or do they like the curvy ones or the branchy ones? And this is an important part of the story, because it's not just about the soil, but it's also about how the cancer spreads to a different part of the environment. And in nature, you sometimes get trees growing in really weird parts, right? Like, how on earth did that tree end up there? Well, sometimes that's exactly what happens with cancer. It ends up in places that we wondered, how in the world did it get there, and why, of all places, did it choose to grow in that area? And like I mentioned before, the brain is a little bit like that too, because the brain has what we call the blood-brain barrier. And the blood-brain barrier is supposed to protect your brain from anything getting across. It's essentially the vasculature in your brain. So imagine that it's kind of like the bouncer of a club. Like, it only lets certain things through because it's a very exclusive area, right? Like your brain. You don't want just anything to go through. So things that are really small can easily go through the blood-brain barrier, but larger proteins cannot. And then there's certain things that your brain actually actively takes up into the brain environment. For example, something like sugar or glucose, right? Like, your brain really loves that, and so it actively takes it and puts it into your brain. Luckily for me, things like caffeine are also something that the blood-brain barrier says, sure, come on through and into the brain area. So, as I mentioned earlier, it's kind of difficult when we're trying to study this question of cancer metastasis, because we don't really want to do them in animals, and we don't want to do them in these simple petri dishes. So what else do we have available? Well, this talk today is going to talk about how we create this blood-brain barrier on the outside of the body using human cells and a technique called 3D bioprinting. There is also another part of our talk that we're not going to cover today, but I wanted to bring up, and that's that we use these computer simulations as well. And by computer simulations, I mean just using math and equations to kind of describe what is happening. And the idea is that we'll 3D bioprint something and make the measurements, and we'll see what happens. But we can also feed these measurements into this computer simulation and get an output that we can then compare to the results. Now, the reason we're doing this is because we would love to come up with a model that helps us predict where the cancer is likely to metastasize. Because imagine being able to make some observations and use this model that then informs the doctors, like, this is where you need to look, because this is the part where the cancer is at higher risk of metastasizing to. What this would mean is you could potentially have early intervention and much more targeted approaches, and both of those things are really important for improving the outcome of cancer. Now, I mentioned that one of the techniques that we use is 3D bioprinting. Now, how many of you have worked with maybe 3D printers in your schools? Okay, I see a couple hands. Yeah, so 3D printing is essentially printing in 3D. So it's kind of a layer by layer approach that you stack up and you create something that is in 3D. And it's made a huge splashy headlines in over the past decade or so, where you now have 3D bioprinters up in space. You've got people that are printing ears and pretty simple organs. And it's really a trend that's really kind of catching and spreading. Now, we're not talking about making full organs. We're talking about making miniature brains on a chip to be able to understand the metastasis process. Now, this is probably a little bit different than some of the 3D printers that you may have at your school. For starters, ours lives in a bubble. And the reason it lives in a bubble is because we're working with real human cells. And the way that you can catch a cold from a friend, these are human cells, so we can potentially infect them or they can infect us. And so we have to do this 3D bioprinting inside of a bubble. Now, all of the magic actually happens in this small area. And so here's a quick video just showing exactly how that process works. So the syringe nozzle is where we store the cells, and then we extrude them out through the nozzle, and we can make any kind of pattern that we want. And we print them into these reactors, and you'll hear a little bit later from Javier why we do these reactors and the role that it plays in keeping these cultures alive. So here you saw a quick demonstration of how we pattern the vasculature in our devices. Now, it's really a challenge when you're 3D bioprinting because you're bioprinting with things that are alive. So you have to really take care to make sure that the cells stay alive before you're going to print them, while you're printing them, and post-printing them. And I always tell people that they really should call it 4D printing because when you're talking about biology, that fourth dimension becomes so important. And that fourth dimension is time. And the reason why time is important is because once you've printed something, you have to then keep that alive. So unlike regular 3D printing where you print something and you can start using it right away, when you're talking biology, you have to give whatever you've bioprinted an opportunity to continue to grow. And like I mentioned, Javier will tell you more about how we nurture and keep it growing after it's been printed. Now, the number one question I always get when I tell people that I do 3D bioprinting is, what on earth are you actually printing? Because most people are familiar with things like plastic or metal, and those are not obviously things that I can print with because cells are not going to grow in environments that are plastic or metal because we are not plastic or metal. So we have quite a variety of different inks that we work with, and I'll tell you a little bit about how they're different. So one of the inks that we work with is a self-assembled bio-ink. And these are made of materials that the body recognizes. So things like collagen, or for example, a blood clot, right? Like you guys have fibrinogen in your body right now, and thrombin, and if you cut yourself, those two things interact and they form a blood clot. And so we print with blood clots because the cells recognize that that's an environment that needs to be remodeled. Basically, they need to fix it, right? Because if you cut yourself, you get a scab, and eventually that scab ends up healing and you have skin that's just like new, right? And so we use that material to help trick the cells into building their own environment. Now, a lot of the things that we build with are soft because humans are mostly pretty soft, right? And so the problem with printing things that are really soft is that they could collapse on each other. So we use what's called support baths to help create an environment that holds the second ink. Now, the way that I like to describe this is, remember when you guys were kids and would go to like Chuck E. Cheese and jump in the ball pits, right? Like you never sank to the bottom of the ball pit because you had sort of all these balls that held you in place. Well, the material on the outside is particulated that way. And so it kind of holds this really soft material in place because of all those little particulates. Now imagine, again, that I've put you in this ball pit and I've put you in different places, and now I've fused those balls together. Now it locks you into place. And so that's what we do with this kind of material. Now we're working with the vasculature, so we want to be able to print the highways that the cancer travels through. And the way we do this is using either a coaxial nozzle, which is just a nozzle within another nozzle, or we use fugitive inks. So the nozzle within another nozzle basically extrudes like a noodle. And this noodle is now a straight channel that we can then perfuse or send liquid in through. The fugitive inks allow us to do much more complicated structures. And we call them fugitive inks because they're only around for a certain time. So we'll pattern the design that we want, and then build around it, and then we'll make it disappear using temperature. And what's left behind are the empty channels of where the fugitive ink was, and that's where we can then introduce the cells as well as the liquid that feeds the cells. Now, I said before that we're not printing with plastic, but we actually are. But we don't use the plastic for the cells, we use it to hold the cells. So this plastic, the silicone, is used to create the reactors that, again, Javier will talk more about. Now, one of the challenges when you're designing a bioink is that you have sort of an engineering challenge and a bio challenge. On the bio side, you have to make sure that it's keeping the cells happy, as I mentioned, right before you print them, as you're printing them, and after. Now, the engineering challenge is I'm actually doing an engineering process, right? So I have to make sure that these inks flow at the viscosity, in other words, that they're just the right type of thickness, that they easily flow, and that they make a structure. But I also have to keep in mind that I have to keep the cells alive while I'm doing this. And so a couple different things go into designing a bioink. One of the things that we talk about is printability. And as I mentioned, that's really looking at how it flows. Because what you don't want is something that is so hard that you end up with these little sort of dots. Now you don't want something that's too easy because then it just flows all over the place, right? So something like water flows very easily, but it's not going to hold its shape. It's just going to be a puddle. So you want something that's just right. So it's a little bit like the Goldilocks problem, right? Like you want something that's just right, not in the two different extremes. Now the other part that we're interested in is, does it hold its shape? So this is what I was mentioning, that once you print it, does it continue to flow and spread, or does it hold its shape? So in this case, you know, you've got a material that kind of oozes out and then continues to spread. So imagine something like honey, right? It flows really easily, but if you let it sit in one area, it kind of ends up spreading. The other one would be maybe something that actually stays its shape. So like toothpaste, for example, right? Like toothpaste, if you extrude it out, it comes out really easy and it holds its shape for the most part, right? Like you put it on your toothbrush and it doesn't really go anywhere. That's the kind of property in a material that we're interested in. Now the other part that we care about from the engineering perspective is, are we getting the right resolution? So a lot of what controls are we getting the right resolution is the nozzle size that we print. So we can take the same material, but if we change the nozzle size, basically the needle that the material comes out of, we can get something that has a really thin feature or much thicker features. And so this is what we call printing resolution. Now, as I mentioned, it's really a balance because you want happy cells as well as good prints. And so from the biology perspective, the challenges that we have there is we, for example, have to make sure that we get an even distribution of cells. Because cells are pretty, I mean, they're kind of small, but they have a weight to them. So they may start off like this, but if they settle, you're going to get them all clumped up together. So we usually have to make our prints that are very quick so that we don't have this settling effect. Now the other thing, of course, that we care about is what we call cell viability. Are they actually alive after you print them? And so things like that nozzle size that I was talking about, while it might be nice if you want a really thin feature, if you have a very small nozzle, you're going to have your cells that are squished and they're not going to be very happy. Now, of course, ultimately, we want to make sure that the materials we're using are not toxic. So while a material, like a synthetic material, might be easy to work with because we can design everything we want about it, it may not be friendly to cells, and ultimately, that's a no-go in printing with cells. Now, the other thing we want to know when we make this big, bulky material is we want to make sure that that it has the right kind of porosity, meaning if we're going to flow liquid through it, we want to make sure that the pores are large enough so the liquid can easily flow through it, and not that it's so tight that we're going to end up limiting the media or the Gatorade that we feed the cells. Now, as I mentioned, printing is only half the battle, and biology is the other half. And Javier is now going to tell you a little bit more about how we keep this biology going to make sure that we end up with happy cells.
Thank you, Monica. So yeah, half of the battle is the engineering and getting all of that to work just fine. The other half of the battle, arguably, probably the hardest part, is the biology. And that is not just getting the cells to do what we want them to do, but also keeping them alive long enough for them to do it. So So if you think about the cells that are in your body, they're organized in a very complex hierarchy. You have cells that build the tissues, that then go together and build your organs. And replicating that is difficult in the lab. And even with bioprinting, we can only get so far in terms of positioning the cells where we want them to be. In addition, we have to think about the cues that the cells normally get from their environment. So, for example, the gels that they're printed in are kind of replicate of what they're normally exposed to in the body. So we have this complex interplay of cells with cells and environment, and all of that has to take time for it to work properly. So in essence, we need a lot of time for them to do their thing. So before I get into how we keep these cells alive for a long period of time, I do want to talk about our reactor and our system. So we start off with a glass slide, and then, as Monica mentioned, we print with a silicone ink, and that helps us build our first structure. And this is pretty much what we're going to use to house all our gels, all our cells, and then also position the pins exactly where we need them. So these pins will connect to fluidic systems later so that we can get the nutrients to flow through. The next step is taking one of the gels that we work with and laying a base layer. So you can think of this as kind of like a foundation that we're going to build upon later. Or you can also think of it kind of as paper that we're going to write on. So once we have this base layer, which by the way is very important, if that first part is not great, our print's not going to work out. So after we have our base layer foundation, we print our sacrificial or fugitive ink on top of it. And one of the cool things, as Monica kind of alluded to, is that our sacrificial ink has this really cool ability to disappear when we want it to. And the way that it does that is, unlike other materials that you might be familiar with, when you expose it to a cold temperature, it melts. And then it solidifies at a warmer temperature, kind of counterintuitive to what you expect with most materials. So this is really great because the gel that we put around it does the opposite, what we expect it to do. It solidifies at warm temperature and it solidifies at cold temperature and it liquefies at warm. And so using those two contrasting characteristics, we can cover our printed design and then use the Fluorescence ability to melt away to leave behind a structure that we then see the cells. And so these structures are kind of like tubes that are in our gel. And they're positioned in such a way that we can connect them to tubing. Okay, so now that we have our device and we have our cells, how do we keep them alive long enough? In your body, you have a very complicated pump system, and that's your heart. And it feeds the rest of all your organs through the blood vessels. We don't have the luxury of connecting a heart to it yet, but we do have a very sophisticated pump system as well that is run and regulated through a computer. And so we'll connect that pump system to a bunch of tubing that then feeds into the bioreactor. So the way that this works, and please don't get hung up too much on the word fluidics, it's really just a fancy way of saying very tiny plumbing. So we have the pump system that pushes pressurized air through the first reservoir. And as it goes through several filters to make sure that it's still clean, it pushes on the fluid in that little tank. And that fluid has no choice but to escape through the feed line of this reservoir, and it leads to the next component of the system, which is our sensor. And this is probably the most important part of our fluidics because it tells us a lot about what's happening. It lets us know how much fluid is passing through. It's telling us, it's actually giving us real-time feedback in terms of if we are playing with how tight those bolts are here. If we tighten them too much, it'll compress the channel and we won't get flow through. And we can actually see this in real time when we're looking at like the computer feed out. And so fluid flows through, it runs through the sensor, and the sensor goes back and informs the pump, hey, lower this, higher this, based on what we need. And we regulate it to about 10 microliters per minute, which is tiny little drops at a time. But over time, that can add up. And so that's why we have these relatively large reservoirs filled with about a cup's worth of water, or nutrients. And so we have a flow rate sensor that's the most important, and it leads into the reactor. And then once it goes through the reactor, it goes back into a collection reservoir, or we call it the exit media. And if we want to, we can do some more metabolic testing, more chemical testing to see what's the health of the cells there. But another way that we can determine the health of our cells after a certain amount of time is to look at them. And the image on the left is our endothelial cells seated on the channel for a little more than 3 weeks. We could have went longer, but we were really anxious to see what they look like. And so all the green you see is staining for the endothelial cells. Specifically the cytoskeleton, the structures that help the cells kind of give themselves structure, support. And one of the cool things is that they're aligned in the direction of flow. So this is really good because if I was to take a blood vessel from my brain and do the same chemical testing and imaging, I would see the same thing, theoretically. Assuming they were exposed to flow. And so another thing that's really exciting is that the cells, given enough time, start to build into the material that they're in. And this is really cool because if you look at the image on the right, you see these tiny little sprouts, and they're forming potentially little blood vessels, and that gives us a good sign that the cells are able to not just interact with each other, but also the environment that they're in. In addition, we can kind of look back and make more complex structures and see how that changes certain things. So here, this is an example of a geometry that was printed, and really we wanted to see that at the end of all of the processing that we do in our experiments, how well does the structure hold up after that? Mainly to see, one, are the channels that we printed circular? Do they actually pass through fluids? And do they maintain the geometry that we intended to have? And in this case, yeah, it worked out. And one of the cool things also is because of the soft nature of our material, it responds very similar to how blood vessels work. So when there's a high pulse of pressure or fluid, you can see the walls expand. And with the increase of flow, you also see an increase of brightness. And that's because the way that we were able to take these images was not by staining cells, but by running these tiny little fluorescently stained beads through. And so here we can just kind of track, and it's just really nice to see that it's responding the way that we would want it to. Oops. And so the next thing, so now that we printed our devices, our models, and we've did some preliminary testing to see, okay, this is potentially going to work out. Can we keep this alive? Is we're going to characterize it and say that we have a model here. So what is a model? So a model is something that is representative of the real world. It's a replica, and it can be scaled up or it can be scaled down. So you might remember having made a solar system diagram in school. So that's taking something that's astronomically huge and making it handheld size that you can take to class. Because there's no way that you can actually bring the sun to class. Or you might have printed a cell, or sorry, maybe you did some 3D printing and you've made your own model that way. In addition, you may have done something that you probably shouldn't have when there was a substitute, and you made my favorite model, paper airplane. So you're probably saying, okay, so this is supposed to be a replica of a plane. how? It doesn't exactly look like a plane. Well, if you take a step back and you think, okay, well, actually the same physical laws that determine if a normal plane will fly apply to this as well. So you have aerodynamics that you have to think about. You have gravity working against you. You have air resistance. And in that way, it's representative. And it's simple. So these are some key things that you want in a model. It's simple, it's representative, and most importantly, actually, I would say, is that it's measurable. So now that I've built my model, does it work? Well, no. In this case, it didn't. Right? So if it flew, I could have said, yeah, okay, I built a good plane here. So now I have to take that and rebuild it and fine-tune the wingspan. So changes to how the plane looks will affect the way it flies, just kind of how like a real plane should work. And so with those three things in mind, being simple, predictive, and measurable, we're going to look to a biological system and say, can we do something here? Can we replicate the complexity that is the blood-brain barrier? And so we have multiple things to keep in mind. We have endothelial cells, we have astrocytes that are another support cell, and they're all working together along with whatever is flowing through the blood system. But how do we actually determine the cells, or how do we actually visualize this? So I kind of showed you pictures of cells earlier, but I didn't exactly say how we were able to capture such small structures. And that's by, the way we do that is by leveraging a technology, and specifically the chemistry of these molecules called fluorophores. And what they allow us to do is, well, what they do is they glow when they're exposed to a bright light. So if you shine them with a high energy light, they get excited. And when they get excited, they release that energy and they give off a really beautiful light. And our microscopes and our detectors are able to pick that up and make these really cool images. So in that sense, so by attaching those molecules to the cells, it's kind of like giving them these little tiny glow sticks. And in that way, we're able to tell them apart a lot better. So here we can see that we have cells from probably 3 different populations. And the way that we can do this can be more specific, so we can actually look maybe at a specific protein in the cell, as we did with the cytoskeleton, or we can just look for certain markers on the inside. So using fluorescence, we can do a lot of science with that, because it gives us a way to detect things that normally we wouldn't be able to detect, at least through the eye alone. And that applies also to permeability. So when we're thinking about things that cross through or don't cross through the blood-brain barrier, we're talking about very small scales, molecules. Even with a microscope, we can't quite see that. So by attaching fluorophores to them, we can detect how intense the glow is, and that gives us information about how concentrated those molecules will be. So in an area where there's a high intensity, we say that there's a higher concentration of those molecules. In areas where there's little to none intensity, that's where the concentration is low. And so we can use molecules that are tagged with these markers in this way to measure one of the most important features of the blood-brain barrier, and that's the function, the barrier function. And this is referred to as permeability. And basically what it is, it's how well the cells are able to keep things from crossing through when they shouldn't cross through. And in this case, we have a nice monolayer of happy endothelial cells, and they're interacting very tightly with each other, helping to prevent unwanted things from crossing through. In this case, we have the little fluorescent molecules I was talking about, and as you can see, nothing's getting past these little suckers. And that's because of this structure called tight junctions. And this is the connection between the cells, and it helps, 1, form the vessel, 2, it also keeps things from crossing through. So if those tight junctions aren't there, or they're very weak, things will leak more and more. as in this case. And this could be indicative of a blood vessel that is not quite mature, or it was exposed to something that shouldn't have been exposed to, something potentially toxic. And so unfortunately, that poor cell did not do his job very well. So what does this look like in our actual experiments? So in the center here, we have a bioprinted blood vessel. And we can take one of those same dyes. In this case, it's a green tagged fluorescent sugar. And we pump it through our blood vessel. And over several time periods, we'll take pictures at the same settings and see how much goes through over time. And we have 3 different time points here with 7 minutes, or 0, or starting point, 7 minutes, and then 15 minutes. And as you can see, as time goes on, stuff is able to leak through, but it's not as disastrous as if there wasn't any cells. And so if we can then do the same experiment with conditions where we don't have cells and see how that compares, or we can do something very hazardous to the cells, potentially kill them off, and then see what that does as well. And that usually forms kind of like the basis of a lot of these permeability experiments. And so this is kind of a representation of what that would look like for the people that can't see the smaller images in the bottom. The next thing that we do is, so now that I've talked to you about how we measure if things go through the blood vessels, I want to show you how we measure how things flow through the blood vessels. And this is through a technique called PIV, or particle image velocimetry. What it is basically in its name, and that is that you're taking images with a high-speed camera of these tiny particles, these fluorescent beads that I kind of mentioned earlier, and we're flowing them through the system, and with those images we're determining their velocity, their change in position over time. And so if we run them through, we can kind of get an idea, as we kind of talked about earlier, if the system is stable, is there any leaks? But also, if we focus in on these beads in particular, we can get an assessment of how they're flowing through the system. Because, as Monica mentioned, the geometry could play a huge role in how things flow through. So if we take a nice little example here, I'm going to show you 2 frames, and we're going to just track the beads as they flow through. If you happen to catch which ones are the fastest, which lane is the fastest, save that in your mind and we'll see if that was correct. So here's frame 1, and here's frame 2. So how many of you guys think you caught the speeders? Raise your hand. I see some people. All right, so keep that in mind. We're gonna see if that was correct, because it's really hard to track that many things all at once. Luckily, we have computers that can help us with that. And we can compare their change in position over not just these 2 frames, but several frames. And we can also do it with more beads running at the same time. So we can also think about this as traffic on the freeway, because that's kind of what we're doing here. The flow rate through our system is 10 microliters per minute, and that's regulated through our sensor, but that doesn't tell us a whole lot. That's how much fluid is flowing through, not necessarily how fast particles are moving. And so, although the speed limit might be 10 microliters per minute, there might be some beads that are going faster than others, and I've already kind of told you that there is. And so, now that we want to determine which ones are faster, we're going to say that faster beads, represented as cars on this freeway or our channel, are different colors. So the red ones are going to be the fastest, blue ones are going to be the slowest, and the medium ones are going to be somewhere in between, in yellow. We also have another metric that we look at, and that's the direction of where they're going. And that's represented by the arrow. So the arrow size tells you how fast it's going, just like the color, but the direction tells you where that bead is going. So this is the results of that little test. So how many of you guys figured that the fast ones were in the middle? Okay, I didn't see anyone raise their hand. It's counterintuitive, especially when you start thinking about cars, like I kind of threw it out there. But the thing to keep in mind here is that, so you have the slower ones in the edges and the faster ones in the center. And I'll talk about why that is and how that applies to our print. So this is kind of what the data would actually look like if we go back to beads. So beads at the edge, again, slower. The ones at the center, faster. And then in this case, the beads were going all straight. So that's not always the case when it's at the edges. So this is what the actual data would look like. And as you can see, there's a lot more going on. And it's not as clear cut. But essentially, the pattern holds true. Slower beads at the edges, and that's because they're interacting with the walls of the vessels. So they're rubbing along, they're bouncing along, and then the ones in the center are pretty much unimpeded, so they're just zooming right through our channels. Just like this. Yeah, if you look really closely at the edges, I don't know if you can from where some people are, there's some that are just kind of like going in circles, so they're just tumbling along. And so this tells us a lot about how the flow is working in our system. And so why is that important? Well, remember, we're interested in seeing how cancer spreads through, and obviously it goes through the blood vessels. And how they flow, or how they potentially will flow through the blood vessel, could play a role in if they're able to attach to the brain vasculature. And so we can take the same idea, same experiment, and run cancer cells through our fluidic system and see if they're able to attach. And so here we took some cancer cells, we dyed them with special colors so that we can actually see them. And all these little round things that are kind of flowing through, those are the cancer cells. This is all within a bioprinted blood vessel. So at the end of it, we stop the experiment, we fix them, and then we see if the cells made it through. So we stained the cells the same way that we did previously, looking for the cytoskeleton. And so here in white, I've indicated where we have the vessel walls. There it is. Oh, well, green and green's not going to work. Luckily, I've highlighted this interesting feature here, and this is showing that the cancer cells were not only able to attach to the vessel wall given enough time, but they also grew along it and then started to penetrate into the wall. And they're likely recruiting some of the endothelial cells in doing this. And so here we've shown that we can actually have a physical model of cancer metastasis through bioprinting. And I'm going to hand it off to Monica.
Okay, so I'm going to bring it back to our original question that I asked at the beginning of this talk, which is, how on earth does metastasis happen? You know, how is it that you can get this kind of cancer breaking through the blood-brain barrier, which I told you, and Javier also mentioned to you, that it's super good at making sure that nothing gets through. And yet something large like cancer is somehow able to squeeze through that blood-brain barrier and grow in a place like your brain. And that's just as weird as, you know, asking, well, how in the world did that tree get there? Obviously nobody planted it there, so how did it get there? And of course it comes to these three things that are the title of this talk, which is seed, soil, and spread. Seeds, of course, are the cancer that look for environments like the soil where they end up then growing and spreading to. And I told you a little bit about how we optimize that soil, right? We use bioinks, and these inks we make sure not only that we can print with them, but that they then have the right kind of material properties, the right biology that the cells can then end up flourishing in. And then Javier told you how we design the vasculature, which is where it spreads through, right? How do we then keep that vasculature, maintain it, flow things through it, and then more importantly, how do we make those measurements? And of course, all of these is to try to create a 3D model that will help us be able to predict and understand brain metastases. Because imagine if you could tell, as I mentioned before, where it's going to spread. This would lead to both early intervention and much more targeted therapies, and so this would potentially help save lives. And you'll see in the movie the effects and the effects that it has on people that have to live with cancer. In both cases, we have one person that is able to get this under control and remission, and then in the case of Hazel, you have somebody that's dealing with essentially the end stages of cancer, when it has spread and metastasized. And what we hope to be able to do is create models that will allow us to help either identify it early enough before it gets to stage 4, maybe at an earlier stage, and have a lot more targeted therapy so that you don't have to then face the more systemic treatments that sometimes cancer patients go through, like chemo, right? That's not in a targeted area. You instead treat the entire body, and then you can potentially weaken it. And, you know, Javier and I were the only ones that presented here, but there actually is a lot more people that are involved in this work, because it really does take a village to be able to handle this kind of big-scale science. And what's really cool about this project is that it involves people from all different areas. You know, Karen, who's wearing the bunny ears here, is actually not a bunny, but a material scientist. And Claire over here is a bioengineer, but she specializes in cancer biology. And then Rick over here, and Morty, who presented. Rick is actually a biologist by training, who then later on became an engineer. And Javier, of course, is our biotech engineer, who helps with a lot of these different models. And it's really interesting because you don't have to have just one kind of educational background. You know, if you're really interested in materials, you know, this is the kind of environment that you would thrive in, but also if you like more engineering and building things like the fluidics that he was talking about, we have a space for that as well. Or, as I mentioned at the beginning of our talk, we do build computational models. So my collaborator at Duke, she just deals with, she doesn't do anything experimentally, but she does a lot of work with computation. So that's really one of the cool things about a project like this in bioengineering, is that it's really multidisciplinary. And with that, I want to say thank you so much for being such a captivating audience, and we'd be happy to answer any questions. Thank you.